according to a British professor, Jennifer Graves, men are on the road to extinction as their genes shrink and slowly fade away. The male Y chromosome is dying and could run out within the next 5 years. A second species of human being could even be born in the future because you do need the Y chromosome to make a male. Professor Graves says, 300 million years ago, the Y chromosome had about 1,400 genes on it, and now it's only got 45 left. So at this rate, we're going to run out of genes on the Y chromosome in about 5 million years. The Y chromosome is dying, and the big question is, what happens next? And to answer the question of what happens next and why something is going on right now, we've asked John Peterson Myers to come up here.
Morning. Moses, thank you. What an extraordinary gathering. And what a sobering set of messages that we've heard and learned about. What you don't know is how deeply this crew here digs into the background of the people who are addressing you. For example, I learned this morning that somehow they found a clip about me, which I had never seen, and they just asked me to introduce it to you. It's about a Senate testimony that I appeared in about a year ago that is about a revolution in science that's underway. A revolution in science which actually has a really hopeful and positive message. And that's what I want to tell you about today. It's related to the fact that in certain communities, including in Canada, just a few hundred miles west of here, there are communities where, in fact, there are fewer boys being born than there are girls, and it appears to be related to chemical contamination in those communities. But if I could have that clip, this testimony is about a key molecule that's at the center of this debate, a molecule that's worth $800,000 an hour.
In the peer-reviewed literature, of the studies of bisphenol A that were funded by government sources, including the National Institute of Health, over 90% of them find adverse effects on animals. In contrast, none of the studies funded by industry report adverse effects. Now, some of you will recall the testimony in 1994 before Congress of the 7 heads of tobacco companies who swore that there was no link between cigarette smoking and cancer. As you listen to industry interpretation of the data on bisphenol A and phthalates, I would encourage you to think about that.
The chemical lobby is one of the most powerful in the world. Since 1976, the U.S. government has banned just 5 chemicals.
Think everything's okay?
Senator, as studies become available to us, we at FDA apply
Studies from whom become
whoever,
available?
if they've been
No, the only studies you're getting right now, have you asked for studies from independent sources?
We don't normally ask for independence.
Then you don't protect the American people if you don't ask for them, if you don't look beyond what's handed to you. Thank you.
So if I could have the slides. This is a battle that's underway, and it's about your health. It's not just about that one molecule, bisphenol A. It's about a series of molecules that we have inadvertently allowed into the products that are part of our homes and part of our lives. But I'm going to tell you some bad news about this, but I want you to focus on the good news, because there is a revolution in science underway today that is telling us there are diseases that are affecting all of our families today that we can prevent. And that's really good news. Let me ask you a couple of questions. How many, if you think about your immediate family and close friends, how many of you have experienced breast cancer within that close circle of people? What about prostate cancer? How about learning disabilities, ADHD? How about infertility? How about uterine fibroids? Okay, I could go on, but I want to ask one last question right now. I want everyone who held up their hand at least once to hold it up again. And I want you to look around and see that almost everyone in this room has been affected by diseases that this science that I'm talking about tells us now at least some portion of those cases are preventable. We don't know, is it 70% or 30%, but we know it is significant. It's preventable, and that's really good news. We've heard a lot about the changes that are underway in the outer world, and they are very big. There are also changes we haven't yet heard about. If I were to go to any tree bark anywhere in the world of a mature tree and take a piece of that bark and give it to an analytical chemist, they would find measurable amounts of persistent organic pollutants in that tree from any mature tree in the world. What about the inner world? What do we find in the inner world? There are big changes underway there as well. If you take cord blood and sample it for chemicals, in this case, a study done in the US, looking for 413 different industrial chemicals, in that cord blood, you'd find an average of about 200 of those, and a total of 271. There hasn't been a child born on this planet for several generations that hasn't been affected by chemicals that are novel to human body chemistry. Novel. They weren't present prior to 1900. We are midstream in a scientific revolution, helping us to understand what that means. Midstream. We're learning that there are profoundly different things underway than we once thought 20 years ago. There are unexpected discoveries about plastics and other things that we've allowed into our lives, things that we thought were safe. But again, focus on the good news. This is providing us extraordinary opportunities to prevent diseases that we did not know were preventable. There are 4 key discoveries in this science. The first is that some contaminants alter the ways that genes behave, and I'll get into that, alter the way that genes behave at truly low doses. A second is that the standards that we have used, the standard ways we've gone about asking, is this safe, is this not? They are blind to the effects that I'm going to describe. The third is that all the tests that we've used to establish safety, they've looked at one chemical at a time, yet all of us have hundreds of chemicals in us. They interact in ways that those tests don't predict. And finally, and this is really interesting, this is one of the most exciting discoveries in medical science for a long time. Adult chronic diseases can be programmed by what happens in the womb. Adult chronic diseases can be programmed by fetal events, by how contaminants and other things affect fetal development. I can tell 3 parts of that story with this one slide. Those are 2 mice. These are experiments done at the National Institute of Environmental Health Sciences in the US, part of the National Institute of Health. Same strain of mouse, same caloric intake for the life of the animals. And there was no statistical difference between how much they exercised. But clearly, the mouse on your right, representative of the experimental group, is very different from the mouse on your left. The one on the right is grotesquely obese. What caused that? Well, that mouse was exposed right at birth to 1 part per billion of a synthetic estrogen called diethylstilbestrol. Diethylstilbestrol is related to a range of compounds that are synthetic sex hormones, like the one I was testifying before the Senate. If that mouse had been exposed instead to 1,000 parts per billion, it would have been skinny compared to the control group. That's a surprise. And obviously, this is a case of early life exposure leading to something different in adulthood. So those are the 3 stories. Low levels matter. A part per billion is not very much. High-level tests, 1,000 parts per billion, predicts what? It doesn't predict the obesity in the animal exposed to 1 part per billion. Early life exposures can cause very severe effects. Well, where does this scientific revolution come from? It actually comes from the genetic basis of many human diseases. And I'm sure most of you are scratching your heads. Wait, Pete, you were talking about environment. You're talking about exposures. How's that rooted in the genetic basis? Well, it turns out that it turns out there are 2 different ways to think about what it means for a disease to be linked to a gene, okay? We do inherit our genes. Genes are what we get from our parents. But here's the crucial thing. Those genes are being turned on and off trillions of times a second throughout our lives. They're chemical manufacturing machines. They're nature's nanotechnology. And for us to be healthy, they have to be turned on or off at the right time. If something goes awry in the mechanisms that are regulating that gene's expression, is what molecular geneticists call it, then you are at risk to your health. What changes gene expression? Well, it does matter what you get from your parents. And genes also are talking to each other all the time. They're sending hormonal signals back and forth, and different types of signals. But it turns out factors in the environment also affect gene expression. Diet, stress, experience, and some, not all, but some environmental contaminants can alter gene expression. Here's a partial list. Bisphenol A, the one I was testifying about, is right at the top. It's become the poster child for this issue. But the list doesn't stop there. It's not endless, but it's significant. And these are compounds that have become ubiquitous in our lives. If you step back for a moment, and you remember the old dichotomy, nature versus nurture, genes versus environment, this new way of thinking changes that. It flips it on its head. Yeah, we inherit genes from our parents, but those factors, diet, experience, stress, and contamination, affect how those genes protect your health. And if something interferes with that, you may have problems. So, how many of you know the Human Genome Project or have heard of the Human Genome Project? What does it do? It does this fantastic work. It uses hereditary signals in human populations to identify what genes are linked to which disease. And so every week, it seems, there's a new publication that comes out. This disease is linked to that gene. Most of us react fatalistically to that. We say, oh my God, did I get the wrong gene? Well, I react very differently. I say, what lab in the world is looking for environmental factors that interfere with that gene's expression? Because that gives us an opportunity to intervene. Now, it may not turn out to be the case, because not all disease is going to be linked to environmental factors in this way. But what we're learning is that a significant fraction of the human disease burden very likely is. We used to think, as I said earlier, that chemicals act by themselves. That's how all testing is done to develop the standards that protect you today. But now we know that chemicals interact unpredictably with one another. I can take a mixture of chemicals, any one of which is below a level sufficient to cause an effect. I put that mixture together, and they can cause big effects. And chemicals also interact with social factors like stress, poor diet, to make vulnerable populations even more vulnerable. We used to think only high levels mattered, that most of us didn't have to worry about this. It was only the people who lived downwind of a chemical plant. We now know that levels that we couldn't even measure 20 years ago can be important. So this product, polycarbonate plastic, is made out of that molecule, bisphenol A, and I could go on at great length about this. It's used in so many products. If I measure any one of you, we're going to find bisphenol A in you. 93% of Americans. I don't know what the number is for Canada. The levels are pretty low, okay, by the standards of old toxicology. They're in the parts per billion. And old toxicology would have said, who cares? I want you to think about what it means to have a part per billion. Let's take a drop of water out of one of those polycarbonate flasks, and just for thinking about it, well, let's think about a part per billion first. What is a part per billion? Well, one pancake in a stack of pancakes 4,000 miles high. Who cares? One pancake. Well, but there's another way to think about it, which I leapt ahead of myself for. If you take a drop of water out of one of those polycarbonate bottles with a concentration of bisphenol A in it at 1 part per billion, how many molecules of bisphenol A do you think are in that drop? Guesses? 132 billion molecules of bisphenol A are in that one drop. And if you have a compound that's an incredibly powerful synthetic estrogen that interacts with genes that are under the control of estrogen, you've got problems. Low doses of bisphenol A are now linked through laboratory experiments and experiments with human cells to a range of today's diseases. The certainty of the science varies among them. The newest addition to that list is heart arrhythmias, which I'll talk about briefly next. For people in this audience, we used to, until quite recently, we used to think that this science was mostly relevant just to things that happen in the womb and then affect adult health that way through growing up. We now know that bisphenol A, because it causes heart arrhythmias, is linked to the risk of heart attacks and also to type 2 diabetes. BPA also, bisphenol A, interferes with the standard treatments for prostate and breast cancer based on the same types of tests that are used to develop those pharmacological treatments. One mouse slide. It's long enough before lunch to show you. This is the inside of a normal mouse. Good, healthy kidneys. Bladder. It's a male. It's got testes. What would happen if I were to expose this mouse in the womb to bisphenol A, a really low level, 20 parts per billion, and let it grow up? This is the control mouse. Here's the experimental mouse. If this mouse had lived another day or two, its bladder would have exploded. Its kidneys already are necrotic. They aren't working. This is urologists' first animal model of what is an increasingly common problem in people called obstructed bladder syndrome. And the scientists who do this work now understand, at least mechanistically, what's happening. If you think about the tube that connects the bladder to the outside world, what you pee through, it's called the urethra. There's a part of the urethra that when exposed to bisphenol A during development in the womb, it alters the control mechanisms that govern when certain genes can be turned on and off. And as that animal is aging, and it's responding to the differences, to the changing hormone levels in that animal, which happens as we all age, those genes are responding differently than they should have. Instead of staying open, they're gradually constricting until the urethra is completely blocked and the animal can't pee anymore. That's caused by a short exposure to bisphenol A during fetal development. So there's some bad news in all this. We have made some unfortunate design decisions about the materials we have allowed into common consumer products. And the tools that we have been depending upon, epidemiology and toxicology, are blind to the types of effects that I've talked about. And I can explain that at length if people want to talk about it later. But there's really good news here, actually. A disease that is under genetic control is actually one that may be caused by the sort of genetic hijacking I've been talking about here. We will have the opportunity to prevent many diseases we didn't know were preventable under the old way of thinking about what it means for a disease to be linked to a gene. We can do a much better job. The regulations that are supposed to protect us today are in the scientific Jurassic. They don't even begin to reflect this new science. Canada actually has moved, the first country in the world, to take action on babies' exposure to bisphenol A, and I thank you for doing that. It's fantastic. But we've got to do a much better job. We can. The science is telling us that we can. We've got some challenges ahead of us. There's lots of elegant science to be done yet to make this even more certain. I think we already know enough to begin to implement changes in how we design some of the common consumer products I've mentioned. We can develop better health standards. And we can give better design signals to an emerging group of chemists who call themselves green chemists, who are actually taking on the responsibility for the toxicology of the products that they're producing before they make it in the marketplace, before a compound like bisphenol A is worth $800,000 an hour, making the debate over it very intense. If you look at today's epidemics, every one of the disease conditions on this list is linked, not certainly, but plausibly, to the types of biological mechanisms I have mentioned today. We can do a much better job. We can help prevent some of these cases by responding to this new information, this revolution in science that is before us. It's really exciting. How many of you know where Bentonville, Arkansas is? How many of you know why Bentonville, Arkansas is important? Okay. Bentonville, Arkansas is the home of Walmart. So I gave a much longer version of this in Bentonville last week, a week ago yesterday. I've been invited back next week because we're going to get bisphenol A out of can linings. It's in 80% of the cans that are on supermarket shelves today. And that's how most adults are exposed. And it's also how a lot of fetuses are exposed because their moms are eating food that comes in cans. So after I met with the executives, I went to the flagship Walmart there in Bentonville. And I walked into the store. I walked into the section for kids. And there were dozens of products prominently labeled Bisphenol A free. I was so excited because I could look at tangible results of work that scientists like myself have accomplished. It's a start. We've got a lot more to do. We've shown that we can do it. We can make products that are much safer, and we can help reduce diseases that currently affect many of the people in this room. Thank you very much.
John, I do want you to help me with the narrative that I was sort of trying to build, which is what is the impact of all of that on the future of the male? You touched on this community 200 miles west of here. We're talking about Sarnia.
Talking about Sarnia there,
right.
Yeah.
Can you tell the folks about
Sarnia? About 10 years ago, maybe a little less than that, the people of Sarnia, which is a Cree community, and lives downwind of Canada's major industrial chemical facility, they realized there weren't enough boys in town to field a softball team. They couldn't do it. And they brought in some epidemiologists to begin to ask, what's happening? And they discovered that there are a number of cases in the developed world. We're not talking about the developing world. We're talking about the developed world. A number of cases where there have been chemical accidents. Seveso, Italy in 1976, a big dioxin accident. There's a Russian pesticide plant that blew up years ago. And in each of these places, there was a dramatic drop in the numbers of boys being born. Now, science isn't certain about confirming that those boys are missing because of chemicals, but that's what it looks like. And actually, if you look at Canada's data, or you look at US data, or you look at Northern European data, there is just as we saw missing girls in the developed world, we're seeing missing boys at the population level. Small percentage, but large numbers. I think the number is about 300,000 over the developed world. And right now, there are multiple explanations for it, but because of these incidents where we see chemical accidents or high levels of certain types of chemical exposures, we believe, the scientists who work on this believe that males are more vulnerable to some of the chemicals in them than the females are. So that's
But isn't the premise that the male fetus is inherently less robust?
To certain types of chemicals, yes. How many of you have heard of phthalates? Phthalates are a plasticizer that's used in a lot of things like rubber duckies. Buy Slow Death by Rubber Duck. It's a great book. It's upstairs. It's by two people from Toronto. Phthalates are anti-androgens. They interfere with the action of the male hormone. They are ubiquitous, just like bisphenol A is, and they're associated in rats and mice. We can cause something called a phthalate syndrome with phthalates by exposure in the womb, and it leads to reduced sperm count in adulthood. It leads to some birth defects of the male reproductive tract. It leads to undescended testes, a class of compounds, a class of effects, which we now are seeing in people in something called the testicular dysgenesis syndrome. And so we know that males appear to be more vulnerable than females to that sort of exposure.
And this alarming loss of genes on the Y chromosome that Professor Graves talks about.
That was news to me.
It was?
Yeah.
All right. Okay. Thanks.
Great, thank you.